Akash, you can start.
Okay, sir. A good evening, good evening to all the participants. I, Akash Shirsagar from the team Just Agriculture, cordially welcomes you all to the fifth day of 1 week faculty development program on effective research methodology and publication, jointly organized by Just Agriculture, the magazine, and SGT University, Gurugram, Haryana, which is ICR accredited university. Today we have with us Dr. Vinod Saharan sir for the session. But before moving forward, our session, I would like to give a brief introduction about him. Dr. Vinod Saharan has completed his master's and PhD from CSC Haryana Agriculture University, Hissar. Our speaker started his professional career as a visiting research fellow at Department of Biotechnology Engineering, Ben Gurion University of Negev, Israel in 2003 to 2004. Later, he worked as a postdoctoral fellow at Department of Plant Genetics, Agricultural Research Organization (ARO) Volcani Center, Israel in 2007 to 2007. He then joined as an assistant professor at MPUAT Udaipur, Department of Molecular Biology and Biotechnology, and now Dr. Saharan holds the position of associate professor and head of Department of Molecular Biology and Biotechnology, MPUAT Udaipur. His awards and recognitions it includes postdoctoral fellowship in 2006 awarded by Department of Plant Genetics Agricultural Research Organization ERO Israel under Professor Aaron Zelser. The second award is Israel Government Scholarship 2003 to 2004 awarded by Advanced Research in Department of Biotechnology Engineering Ben Gurion of University of Negev, Beersheba, Israel, co-sponsored by Ministry of Human Resource Development India, awarded with certificate for appreciation by Lions Club of Mayor in collaboration with MPUAT on a Teachers' Day, 5th September 2013, and many more. He has acted as a principal investigator of various external funded competitive and project under DST, DBT, Ministry of Agriculture and RKVY and overseas research project funded by DST New Delhi and CISC MSRT Iran. Dr. Vinod has been an inventor of nearly 4 patents. He is the reviewer and editor in various reputed national and international journals. He has guided 17 MSc, 7 PhD scholars, and 1 postdoctoral fellow. Under his working profession, our speaker has published numerous research papers, book chapters, and articles. So now, without wasting much of time, I would like to welcome Dr. Vinod Saharan to take over the virtual podium and start his session. Yes, please, sir.
Are you getting my voice?
Yes, sir.
You're audible.
Okay, fine. So let me start my presentation. Uh, Thank you for your introductions of mine. And please allow me to share my screen. I'm not able to share. I'm not getting option to share.
Sir, we have given you the rights to share your screen.
No, it's not possible, I think. There is no option I'm getting to share my screen.
So do one thing, you can just share your PPT with us. We will share it by us, sir. And you can just go with your talk.
Okay.
Sir, you are a co-host and there is a green color button of share screen in the lower bottom. Please check.
I click already, but after clicking, I am just seeing entire screen, window, and
Yeah,
Chrome.
you have to select the entire screen.
Yeah, I have already selected. Now I'm clicking on that icon, but I'm not getting to my window.
Sir, you have to switch to the PPT now. Please open the PPT. Then your screen is visible to us. Or you can mail us your PPT or WhatsApp us. We can share from our side also.
You are getting my PPT now?
No, not yet, sir.
Not. That's why I'm saying there is an issue.
Sir, you can share us by Utkarsh ma'am, please coordinate.
Yes, sir, I've already shared him the.
Please mail us your PPT or WhatsApp as.
This is too much.
Sir, kindly send your PPT to my WhatsApp, or I have just dropped you a message with my email ID. You can just share it with.
Okay. I didn't receive your email. It is not there.
I have dropped you a WhatsApp, sir. Sir, have you shared the PPT?
Yeah, its size is very big. It takes time, ma'am. It's still uploading.
Okay, sir. Okay. So by the time you are sending us the PPT, you can just go with your talk if possible.
Okay. I sent mail to you.
Okay, sir.
Sir, you can start. Screen is shared.
Okay, thank you very much for inviting me for this lecture. It is initially some problem, but anyhow, let me make the presentation. So I'm going to talk about the biomaterial based nano invention for agriculture. And as sir is telling about my intro, I am currently associate professor at the University of MPUT Udaipur. That is one of the agriculture university in Rajasthan. And I am heading Department of Molecular Biology and Biotechnology. And also I have a lab on working on nano research in agriculture. And we are working since last 15 years on the nanotechnology in agriculture. And initially, I trained in Israel for my PDF and my PhD research in ARO Volcani, in Tel Aviv, and in Bangor University. And in 2007, I joined as a scientist in India. So let me proceed for the presentation. Please change the slide. So my voice is coming clearly to you all.
Yes, sir.
Okay, so as I told you that my place is Udaipur, that is in the south part of Rajasthan. It is close to Gujarat, and we have a very old college. The college that belongs to our university is one of the oldest agriculture college in India, before GB Pant Agriculture University. This is an interesting fact for us. Next, please. So you can see the picture of our college, very historical building we have, very old, and it is started in 1955, the oldest college of agriculture in India. The university is situated center of the city. It is a lake city of India, and I am posted in Department of Molecular Biology and Biotechnology. Next, please. Next slide, please. So I'm going to talk about the nanotechnology, the biomaterial based innovations in agriculture, and I'm heading Nano Research Facility Laboratory. And we have all the basic facility for synthesis, characterization of nanomaterial for our application in agriculture. We have so many students related to the master's, PhD, and also PDF. Next, please. So these are some of the pictures of our department, our laboratories, and as many of us knowing that what kind of instrument that we are needed for nano agricultures, we have instruments related to the synthesis, like a probe sonicator, like inbuilt periodic set for cross-linking. We have instruments related to the measurement of size distribution, PDI, zero potentials of the material that we are synthesizing. And we have instruments like FTIR for checking the interaction between the molecule which we are synthesizing in the laboratory. Next, please. So these are some of the pictures that I told about the instrument that we have. Next, please. So I will talking about two things. One is the basic understanding of nanomaterial, and second part is challenges and uses of biomaterial, the case study in agriculture. Next, please. So first of all, I think those students are now online or so they are listening. They must know the definition of the nano. So nano word is a Greek word which is meaning is drop. That is, in more technical terms, the word nano means the 10 to the power 9, 1 millionth of meter. So if you are going to compare a material to a nano, it is around 10 to the power 9 times shorter than a scale of a meter. Next, please. So there are so many definitions you could find in books, internet, and various the federal agencies have given different definitions up to now, and still the definition is not been universalized among this different scientists and institute. Well, the definition that initially given by the National Science Foundation US, nanomaterial refer to material with the size of 1 to 100 nanometers. This is the normal, we used to heard this definition everywhere. So those material which has size 1 to 100 nanometer is known as nanomaterials. The other definition that is given by the EPA, the Environmental Protection Agency US in 2007, they say understanding and control of matter at dimension of roughly 1 to 100 nanometer, where unique properties make novel application possible. They add something in the upper definition, the definition given by National Science Foundations. They say size is not important. Along with the size, you must have novel property with the material. Then you can say it's a nanomaterial. Next, please. So further, there are so many definitions has been given. One definition is something different what we have learned in the 2.1. The nanomaterials are defined as a particle which at least to one dimension less than 1,000 nanometer in biological sciences. So mostly definition we used to see in the internet or in books, they are related to physical science. They used to say 1 to 100 nanometer. But when we are going to talk about the biological sciences, in biological science definition is different. Definition is where the size range reaches up to 1,000 nanometer. Fine. And why this is happening in biological sciences? Because biological sciences, the biomolecules are itself already a bigger size. Like most of the biopolymer we used to use, we have a cellular molecules, protein, carbohydrate, fats. Their initial size is very bigger. Fine. And they have self-assembled property. They used to come together in a big macromolecules. Fine. That's why their size could be reaches to 1,000 nanometer, and up to that 1,000 nanometer, they are also called a nanomaterial. Next, please. So more definitions. Now, as I told you that different federal agencies has different definition of the nanomaterial. Like in Europe, US, and Australia, they are different. If you're going to go to the European Commission's recommendation of definition, that is 1 to 100 nanometer, and in which they say if you have a particle which volume of half is actually in this range, they are known as nanomaterial. Fine. Like in European Parliament and Council of the European Union, they say that 1 to 100 nanometer size is okay, and there is no threshold amount, maybe 10%, 5%, 100%. They are not specific for that one. Like US, in US say, United States Food and Drug Administration, they give a definition that 1 to 100 nanometer and larger. They can be a larger one, fine, up to 1,000. And there is no specific mention of the threshold amount. Fine. Like Australian government, they are a little bit specific. They say 1 to 100 nanometer and minimum 10% of the whole may be in this size range. So as I told you that the definition is still not uniform amongst the scientists and institute and countries. Different the regulatory agencies of different countries has different definitions of the nanomaterial. Next, please. Like Health Canada, they say 1 to 100 nanometer or larger. Fine. So like Taiwan Council of Labor Affairs, they say 1 to 100 nanometer or larger. Fine. So likewise, Belgian Federal Public Service Health Food Chain Safety and Environment, Belgium 2014, they also say 1 to 100 nanometer, but the content of the material should be 50%. Fine. So I mean to say the definition will still vary from the country to country and agencies to agencies, or where they are going to apply. Whether they are directly apply in the human beings like they use as a food, or they are used in a medical and some other thing. Fine. Next, please. So what we used to call nanomaterials. So what definitions we should learn from reading after so many definitions from the different agencies and institute. So we have to learn like two materials that have external dimensions or internal surface structures with at least one dimension between 1 to 100 nanometer or 1,000 nanometer, with the condition they must have unique characters which relates to the size. So if you are going to consider a physical science, then size may be 1 to 100 nanometer, fine. If you're going to consider the biological science where biomolecules are involved, then size may reach to up to 1000 nanometers, not beyond of 1000 nanometer. And they must have a unique property with the size. If you say I have a particle size 500 nanometer, but it doesn't have any unique property, so they are not considered into nanotechnology or nanomaterial because there is no property with related to size. Fine. So that is very important. Now, if you're going to learn more things about the Indian perspective, then you have to learn the Indian policy on agriculture and nanotechnology. So you have to go to the read the guidelines for the evaluations of nano-based agri-input and food product in India, that is designed by DBT, where you can find the regulatory clearance data, definition, classification of nanomaterial in Indian perspective. Next, please. Next slide. Yeah. So I'm going to read three definitions. One is already clear. The next one is nano agri input product. Now, is defined as the agricultural input preparation containing nanomaterial indented for the application through soil, seed, foliar, drip in crop as well as by other means on crop for purpose of farming. So that is known as nano agri input product, NAIP. And other thing is nano agri product is defined as the agricultural preparation containing nanomaterial intended for the consumption, application in food, feed, and their supplement as well as the nutraceutical delivery. So in agriculture, two type of nanomaterial could be classified. One is the nano agri inputs, the input, the application of fungicide, pesticide, fertilizers, they are coming in this category. Whereas nano agri product, that the product that is going to consume by us, nano feed, like nano protein or nano carbohydrate or some composition of thing which is directly used by us, that is come under nano agri product, NAP. Next, please. So categorizations of nanomaterial according to the nano form of the active ingredient. So nanocarrier loaded with a active ingredient. One. Second is the active ingredient converted into nano form. Fine. According to synthesis, synthesis can be chemical, can be biological. According to that, you can say chemical synthesized nanomaterial and biological synthesized nanomaterial. According to chemical nature of nanomaterial, either nanomaterial organic material or inorganic or composite. According to that, you can divide. According to the degradation nature of the nanomaterial, either they may be biodegradable, they may be a soft nanomaterial, more easy to get regulatory clearance, and other is non-biodegradable hard nanomaterial, which is metallic or other thing which is not able to degrade in the environment. Fine. So overall, nanomaterials are two types. The material itself converted to nanoscale, or the material can be encapsulated into nanocarriers. Fine. So next, please. So in nature, we have so many nanomaterials surrounding us. You can see the picture here. Wind erosion is also making so many dust particles, which may be in nanoscale size, which is also known as top-down approach. And it's bigger particle because of the wind erosions, they become nanoscale particles. Weathering is also doing this job. Volcanic activity, volcano eruptions is a source of natural nanomaterial in the air, and they can stay up to 6 or up to 10 months in the air suspended because of the nanoscale size. Cosmic dust contains so many environmental nanomaterial in the air, and you have a abiotic or biotic uh, reactions happening in the living organism, in a soil, in water. They are also producing so many nanomaterial in the environment. Biomass consumptions is also a big source of nanomaterial naturally. So all together give rise to a aerosol colloidal that's suspended in the air and which are in nanoscale. So we are surrounded with so many natural nanomaterial, and so it is not a new thing that we are learning. It is still in our nature. Next, please. So classification based on the types of materials. Now, first is engineered nanoparticles. Engineered means you are intentionally preparing them artificially with the different techniques like carbon nanotubes, nanodots, wire, and other thing. Incidental nanoparticles preparations, industrial process which you are doing, but that is incidental. You are not preparing it, but they came as the waste material of your process. Natural nanoparticles that we have just read, erosion, volcanic eruption, plant, animal are source of nanomaterials, water bodies, river bed, the water which is coming from the river, you can see at the bed, you can see the small particle. They have so many nanoscale micronutrients. Next, please. So based on the shape and type of nanomaterial, so nanomaterial can be a cavity or a pore. They can be a network or powder form. They can be structured surface. On the surface, they may be present as a dot. They may be a silt. They may be planar surface. They may be a particle in round structure. They may be shell, which is known as nanocarriers. They may be spheroid in structure, spheroid shell. They may be a rod side, tube side, starfish. So different types of nanomaterial can be synthesized or available in nature. Next, please. Classification of nanomaterial based on the dimension. So we can divide four type of nanomaterial based on the dimension: 0D nanomaterials, 1D nanomaterial, 2D nanomaterial, 3D nanomaterial. 0D nanomaterial, all dimension at the nanoscale. As I told you in definition that at least one dimension must be nanoscale. Fine. So if all dimension nanoscale, it is known as 0D nanomaterial. If two dimension at nanoscale, it is known as 1D. If it is one dimension at nanoscale, it is known as 2D. If no dimension at nanoscale, all dimension at macro scale is 3D. But these 3D particles or material made up of 0D nanomaterial. If your spherical shape or oval shape nanomaterial, which is all dimension are 0, aggregated together, so they are known as 3D nanomaterial, which is an example of diamond. Next, please. So if you are roughly compare the nano size, you can compare. If you are holding a tennis ball in your palm and you compare this size with the earth size, the tennis ball known as a nanomaterial. So this much small size that nanoparticle have. Next, please. Next, please. So size scale of biological system, that is very important to heading towards the plant system. So if you see the size of cell, the plant cell, pollen cell, animal cell, average bacterial cell, there lies from 40 micrometers to 1 micrometer. A bacterial cell size is 1 micrometer, 1000 nanometer. So most of these cellular system like plant cell, pollen cell, animal cell, or bacterial cell, they are above 1000 nanometer. Fine, they are not coming in a nanoscale. But going to go to biomolecules by which they are built of, they are in nanoscale. S2 size of the hydrogen atom, S2O, catalase, sucrose, protein, DNA, ATP, protein, ribosome, gene, atom, cell membrane. Their size are ranges from 0.1 nanometer up to 25 nanometer or 200 nanometer. Fine. So it means that our cellular system is made up of so many nanoscale biomolecules. Fine. And that's why their efficiency of working is very surprising. They are managing each and everything in the cellular system. Fine. So this is the scaling of biological system, and by this scaling we can learn or that why we use 1 to 1000 nanometer scale of nanomaterial. Next. So normally in biological science we used to say 1 to 1000 nanometer if any particle having a size and they have have a to learn why nanomaterials are so. Please go back to the last slide. Bring back last slide. Yeah, here. So if you see the all the natural opening or passes in plant cell, you could see the size range from zero point 0.1 nanometer to 1000 nanometer. Cell wall size is around 10 nanometer. Cell membranes are around 1200 nanometer. Like vascular system is also kept in 1000 nanometer size. Cuticles, stomata size is kept in 1000 nanometer. Started from 300, 400 nanometer. Like hydrothores or lenticels, their size is below 1000 nanometer. It means all the natural opening or passage, if you're going to supply something to a plant and their size kept in 1000 nanometer, they can easily go through these passages and opening. That's why nanomaterials are more effective compared to the bulk material that we used to use. Now, this is very inspiring, and this is the reason that in biological sciences we used to define the nanomaterial size range from 1 to 1000 nanometer, because at that size scale they have unique property to crosses the natural opening and passes of the plant cell or biological system. Next, please. So biological system itself is a nano world. Why? Because nucleic acid synthesis, photosynthesis, protein synthesis, signal transduction, natural opening of cells all work at nanoscale. If you're going to for photosynthesis, in photosynthesis you have chlorophyll, you have grana. So they absorb the light from sun. They are in nanoscale size. Their surface area is so much that they can absorb a lot of light from the sun and convert this physical energy into the chemical energy. Likewise, you have protein synthesis there, where you bring amino acids together with the mRNA and rRNA. They assemble in a nanoscale and precisely do the translation of protein. Likewise, other biological processes are also following, and they actually the synthesis process occur due to the nanoscale biomolecules. Next, please. So what we conclude, biological system is a nano world. Almost all biological events, pathway activities come through a nano, and all passes opening exist are in nanoscale. Nanostructure may more interactive to cellular system as compared to bulk or larger structure. That's why nanoparticles, nanomaterials are more effective. They have more biological activities. Next, please. So if you're going to learn about the physics of the nanomaterials, the so-called quantum size effect describes the physics of the electron properties in solid with a great reduction in the particle size. So this effect does not come into play by going from macro to micro dimensions. However, it becomes dominant when the nanometer size ranges are reached. So when particles are in nanoscale size, they are justified by the quantum theory, fine, in which the electromagnetic interaction are dominating, gravitational force are not dominating, and you can understand by example of such effect is superconductivity, which is a phenomena whereby charge move through the material without meeting any resistance. So therefore the electrons are more movable in nanoscale particles. They are more interactive to the environment. That's why the nanomaterials are more active. Fine. This is you can understand by the quantum effect. Next, please. So why nano-sized structures are more interactive for biological sciences? Say, for example, you have a block of 6 centimeters square. You break it into the smaller pieces. Now the size increases to 12 centimeters square. Now again you break in more small pieces. Now size increases to 24 centimeters square. When surface area is increasing, more molecules come on the surface. When more molecules are on the surface, they are more interactive to the environment. If you have some fertilizer with you press it, you make in a powder, you make in a super powder, you bring into a nanoscale size. So surface area is more, they are more interactive to the plant cells. That's why they are more effective. Next, please. So this can further understand if you have more molecule on the surface, molecules and interaction and molecule and their electrons are more free to interact because their pulling power is less towards the center part because more molecule on the surface. Once your electron is free to interact, then your molecule will give a nice response to in during the interactions. Next, please. Next, please. I think this is I already explained. Next, please. Yeah. So how we synthesize nanomaterials. So there is two approach. One is top down, in which the bulk material, bigger size materials can be cracked down in a smaller size. This can be understood by our traditional kitchen, in which we used to prepare the chutneys and other thing by grinding the bigger particle and smaller particle, and they are become more tester for us because their surface size is increased and their test, when we are going to put in on our tongue, their test is so fast because they can more interact to our test buds because their size is smaller. Another is bottom up approach, in which the more smaller size, less than nanomaterials, you bring together those ionic forms and convert into nanoscale. That is known as bottom up approach. The classical example of bottom up approach is the DNA replication, protein synthesis, and other thing. Next, please. So what is the instant application of nano in agriculture? Now we are hearing since last 3 to 4 years about the nano fertilizer. So IFCO nano urea is there in the market now. There are more products are coming in the market. So nano fertilizer is going to be play an important role in agriculture in India and also in globe. So what is the definition of nano fertilizer? Nutrients converted into nano size or nutrient coated encapsulated into nano carrier for higher nutrient use efficiency, slow release, control, or target release. That is known as nano fertilizers. Nanofungicides, again the same, either fungicide or pesticide or herbicide itself converted to nanoscale, or they may encapsulate into the nanocarriers for enhance their efficiency. That is no nanofungicide or pesticide or herbicides. Now in future we will have some multi-targeted nanocomposite. What are they? More than one active nanoform or encapsulated nanoform to perform multiple tasks in plant growth as well as plant protection. So a capsule which is maybe a nanoscale size, which contains the macronutrient, also pesticide or insecticide. So they may perform multiple tasks. Nano coating, edible coatings. Now, starch, cellulose, biopolymers, calciums, copper, hexanol based nano coating for minimizing the post-harvest losses. Now, this is nano coating to prevent the post-harvest losses. Normally, when we go to the market and purchase the apple, you will see the wax on the apple to protect it from the losses of the weight and moistures. And you can, when you are going to scratch out, you can feel the wax is coming in your nail. So in coming features, nano coating, edible coating will be there. You are not able to see the coating because they're in nanoscale size and they are safe to use. Next is nanosensors. Nanosensors can play an important role in agriculture in the future. Nanoscale entities act as a sensor showing light spectrum changes, color for physiological or biochemical changes to disease, nutrient deficiency, mostly quantum dots, gold nanoparticle, that is for detection of virus and fungal disease in the field. So nanosensor, that is known as super nanosensors, will come soon in agriculture. Next, please. So what is the challenges for agriculture? Now our population is increasing. By 2050 we are going to reach about 9.7 billions. Other problem is global warming, and all together give rise to a food security, and we are looking for almost double food in 2050. The resources that we have at present, they are not enough to feed us in 2050 because of the problem of high population and global warming. Now global warming is inducing because we are using huge amount of agrochemicals. Our air, water, soil is polluted. And please go back. So we need to sustain our agricultures. Fine. Therefore, we are looking for a new technology where we can suppress the global warming to not allow the environment to change, because if environment is going to change, then they may change all the host ranges of the pathogens and disease causing organisms, and then you have to use more agrochemicals. Therefore, we are looking for a solution which can sustain our agriculture by decreasing the application of agrochemicals in agriculture. Next, please. Next, please. So what is the savvy notation for sustaining agro ecosystems? So we have to look towards the biogenic materials, biomaterials, biologically derived materials, that is carbohydrate, protein, lipids, or their conjugates. So because means these are some of the material which is present in the nature, and we have to extract them and use in the agriculture because they are biocompatible, biodegradable, wide functions they have, they act smartly, fine, they are non-toxic and biologically degraded. They act as an elicitor for plant. They are very good antioxidant defense system for the plant. They can act on demand, on command, slow control or target activity. Next, please. So biomaterials have unique properties for nanotechnology customizations. It has n number of functional group available, which can be used for loading of any active ingredient, highly compatible for structural and chemical modifications. We can change the size of this biomaterial easily, shape, dispersity, surface modification, surface charge, and elemental conditions. Next, please. So we are exploring in our research a chitosan, a aminopolysaccharides. We are exploring thymol, which is from the thyme plant. We are exploring saponin, which is present in most of the vegetables, and we are exploring some signal molecule like salicylic acids to make the novel nanomaterial for application in agricultures. Next, please. So mostly we are exploring chitosan, which is a biopolymer, amino biopolymer, and which is extracting from the bio waste of seafood industries, from exoskeleton of crustacean class of living organisms. It is also present in mushroom. The chitosan is similar to chitin and cellulose besides its NH2 group. In chitin, it is NHCO CH3 group. In cellulose, it's OH group. But in chitosan, is NH2 group. That's why it has a biodegradable properties. It is biocompatible, non-toxic. It acts as a plant elicitors, induce plant growth and defense. It has a good antimicrobial activity, but it is not soluble in water. That is the biggest issue with the chitosan besides other positive properties. Next, please. So we have explored chitosan like anything. As I told you that these biomolecules, biopolymer has active functional site, like it has an NH2 group where we can add any active ingredient. We can add macronutrient, micronutrient. We can add fungicide, pesticide on those sites, which is act as a port, just like a helicopter having a port side where you can attach the arsenals. Uh, so so if you are going to explore these biopolymer, they can carry so many things. So they act as a nano carrier for active ingredient or agrochemicals in agriculture. Next, please. So chitosan normally is a linear biopolymer, and when you are putting in the water, they are not able to dissolve. So therefore, you have to add acid. So this is acid soluble. In acid soluble, the NH2 group become positive charge, and it's soluble in water. But the acid soluble chitosan is not compatible for application in plants because the pH is less than 3, and which is toxic for the plants. Therefore, we must have some techniques to make it colloidal or soluble in water without using acids. So therefore, we converted these chitosan into nanoscale chitosans. and use as a nano carrier for supply of macronutrients or other agrochemicals. Next, please. So we convert the linear cytosine biopolar into the polycationic cytosine in nanoscale by using ionic gelation method. Next, please. So here linear cytosine can be converted into the nanoscale material by using ionic gelation methods, and these nanomaterial can act as a very good carrier for agrochemicals to supply the slow release of macronutrients or pesticide or fungicide in agriculture. Next, please. So you can use these cytosine nano carriers for encapsulation of zinc, encapsulations of copper for application in agriculture as a fertilizer. Next, please. So there are so many techniques to characterize these nanomaterial. You can check the size distributions, number intensity, polydispersity index, what is the charge on the surface, and what is the interaction between these molecules, like what is the interaction of zinc to the chitosan, what is the interaction of NH2 group to the other functional group of agrochemicals. So you can do analysis using various techniques like DLS, FTIR, NTA, TAM, SAM, and you can also do the elemental composition, how much amount of zinc is there in the nanoparticles. Next, please. So these are some of the pictures of these nanomaterial based on the chitosan. Chitosan act as a carrier for various active ingredient for application in agriculture, mostly in the cereal crops, for disease protections for the growth. Next, please. So here you can see the pictures where we have used copper, zinc, silica, and salicylic acid into the nano carrier of chitosan for application in agriculture, for disease, for protection from the fungal disease, for the higher growth, for supplying of nutrients. Next, please. Next, please. Next. Next. These are some of our applications. Mostly these nanomaterials used as a seed treatment, as poly applications. So when you want to treat the seeds, they increase the seed vigor index, seedling growth, shoot length, root length by acting on the seed mobilizing enzymes in the plant. Next, please. So here we have used one of the patented technology that in which we have make a slow release of salicylic acid through chitosan nanocarrier in a maize plant to control the disease, fungal disease. And this system is not only effective for the disease control, but also for other abiotic stress. Next, please. So this is one of the slow release technology of salicylic acid that we have a patent in our lab. Next, please. This is one of the zinc encapsulated chitosan nanomaterial in which we have encapsulated zinc into the chitosans and use against CLS disease in maize, and also it acts as a fertilizer for plant to supply zinc through foliar application. Next, please. Next, please. These are some of the results of the field, and this is another very good technology that we have. Normally, the oil is not able to mix in water, so people used to use surfactant into the water. We make a nano emulsions, plant-based nano emulsion, in which we use thymol. Thymol is a essential oil component of the thyme plant, which has a great activity against the fungal bacterial diseases. So we convert this thymol into nanoscale through emulsion technology using the plant-based surfactant, which is a patented technology that we have. It can be used for polyhouse grown plants. It can be used for the cereals. It can be used for any other crop. and it is plant-based. Nothing is synthetic here, and it is also biodegradable, so it can supply nutrient and it can protect your plant from the disease. Next, please. Next, please. This is the same that I explained. So our lab has a 4 patent, as I told you about the different applications from nano carrier cytosine to the emulsion technology to also the control release of urea. Recently, we received a patent on nano-based urea formulations. Next, please. So our lab is funded by DST and DBT. We have a quite good funding till date, and we have a good collaboration with other institutes like Indian Institute of Maize Research, Ludhiana. We have a collaboration with the CFAT Ludhiana, which is working on post-harvest losses. We have a collaboration with TNAU Coimbatore. Next, please. So these are, I acknowledge my work to my students, and they are doing and they have done a great job for the nano agriculture in our lab. Thanks for your patience to listen to me. If you have any question, any interactions, if you have any inquiry, go ahead.
Thank you so much, sir, for your informative talk. Thank you once again for giving your valuable time for the session. Thank you, sir. Thank you, dear participants, for joining the session and for patiently listening the session. Thank you and have a wonderful evening.
Thank you, sir.